Good morning, everyone. This is Mr. Y. Today, this is going to be the first small video in a three-part series I have that looks at dissecting experiments that have actually occurred in biology in real life and trying to help you in getting comfortable with identifying the different aspects of those experiments. So again, this is only going to be the first of a three-part series. Each one will deal with a different experiment, but in each video I will go through helping you identify the different parts of that experiment and how it was run. There will also be videos embedded with each of these to help assist you in kind of understanding how the experiments actually worked. Again, these are real experiments that were done. Some of them were really important in biology, but they're all fascinating. And again, they're all real. So with each experiment that we see, we hopefully are able to identify the various anatomical features of that experiment, how the different aspects work together. There is a video here that you should have watched already. If you haven't, you can watch it at the end of this about, for instance, how you can write a successful hypothesis. I've also linked the video down here in this little link there, which if you download this PowerPoint somehow, if I've given you access to it, then yeah, you can do it that way. But I will also put the link for this video in the description, and you could also just search by the name. It's right there. But with any experiment, there's always certain features we want to look at. Usually the easiest things to start with identifying are the independent variable and the dependent variable. And while I'm not giving you the definitions here per se, I did that in another lecture, what I am doing is giving you ways in which you can help to identify them relatively easy. And I'm going to go through this with each of these three videos. So when you're looking for what the independent variable is, you simply want to ask, what is being changed by the researchers on purpose? What are they manipulating? Sometimes this is called the manipulated variable. So what is actually being changed on purpose during the experiment? In contrast, the dependent variable, that's what you are actually measuring or recording. Are you measuring in seconds? Are you measuring time? Are you measuring, a sound in decibels? What is it you are measuring? Are you measuring growth in inches? Whatever you're recording precisely is your dependent variable. So what you change is your independent variable, sometimes called the manipulated variable. And what you measure, what you record, is your dependent variable. This is the dependent variable is sometimes called the responding variable too. Then you should be able to determine what is your experimental group and what is your control group. Now the experimental group is really the group where the changes are made. Usually this is the group you're the most curious about, but it's where the changes were actually made in contrast to the control group, which is what we're comparing the experimental group from. So where you make the changes, that's your experimental group, and you have to usually have a comparison to see what would have happened if you hadn't made those changes. That's the control group. Constants are kind of self-defining. These are what are being kept the same between both the experimental group or groups and the control group or groups. What is it that you must make sure is the same? Do you have to have the same amount of time, the same amount of space, the same location, same lighting? All these different things come into play. So constants are those things that are being held literally constant. And then of course, what would the hypothesis be? We're going to talk about this, and I'm going to save that part for last because it does tend to get tricky, and that's why I wanted you to watch this video about how to write a strong hypothesis first. But essentially, the video here shows three different styles. The style I'm going to use is actually a merging, a blending of all three styles that are put forward in the video. So I'm going to call my version a blended hypothesis, which is really just taking the three styles that this lady in the video shows and merging them all together to give you a nice, complete, and very robust hypothesis. And the three styles she'll show you in the videos are what she refers to as an if-then, and then a causal, which usually use the words will result in, and a comparative type of hypothesis. So my blended style will actually use all three of these in one. So there is another video. Again, I've embedded it here in the PowerPoint. There's a link down at the very bottom. The video should also be linked in the description as well. Now, the basis for some of this experiment that you're going to see, it was actually started way back in the 1700s by a scientist whose name I'm going to mispronounce. He was an Italian scientist by the name of Lazzaro Spallanzani, I'm sure I messed it up, who observed that bats could fly about freely in a dark room, even though it was pitch black, even if they were blind, and avoid obstacles. That was the initial observation. A little later on, another scientist, whose name I'm also going to mess up, by the name of Charles Urin, found that when bats' ears were filled with wax, the animal was basically helpless when it was flying and tended to collide a lot with obstacles. And this was eventually followed up many, many years later in the late 1930s by two gentlemen from Harvard University by the name of Galabos and Griffin, who were actually one of the first sets of gentlemen to actually get their hands on what was newly discovered as an ultrasonic detector. The idea that there were sounds that were beyond the human ability to hear, and these ones were high frequency sounds. And their ultrasonic detector came from a gentleman by the name of William Pierce, and they were able to prove that bats emitted ultrasonic noises that you couldn't normally hear. So you might think the bats were silent when they were in fact were doing a lot of vocalization. It's just they weren't within our range of hearing up to the point until we had these ultrasonic detectors. And that this ultrasonic sounds that they emit is how they were actually able to use echolocation to fly about. So this is part of the experiment we're going to be looking at today. And again, you can watch the video on the right hand side over here, or watch it from the description. But yeah, take a look. It's not the best video quality, but it does show in essence what they were doing, how they were testing the bats and how they did various things. So make sure you watch that before you go on to the next part. It'll make the next part make a lot more sense. Okay, so the basic gist, though, of the experiment the two gentlemen from Harvard accomplished in the 1930s, late 1930s, early 40s, is that they would put bats basically in a black room, and in this room they had a whole bunch of obstacles hanging down. They didn't have obstacles so much like this. What they did is they originally had wires just strung straight down, and they found that this created a nice experimental chamber for testing the bats that they were looking at. And in some cases, if you look at more modern versions of this test, they use nettings, or like you can see in this picture, they use like these little foam things that kind of swing in the way. The original test, though, was done with just wires as obstacles. The bats, they figured out, could fly about just as the original scientists had observed very easily, no matter the light conditions, whether the bats were truly blind or not. Some bats aren't blind and some bats are, but they could easily navigate the room without any collision. They could land perfectly fine, go from wall to wall. They could land on the walls, and they had no problem navigating the room, whether the lights were on or off. The bats themselves, however, were also made to fly about the room with different parts of their sensory organs on their head temporarily blocked, disabled. They didn't like permanently injure the bats, but they figured out ways to disable certain parts of their sensory systems. So in some cases, they were temporarily blinded. And if you watch the video, you'll hear the gentleman talk about how they used some kind of, I think it was an ink or a glue or something. They just glued their eyes shut so they couldn't see. In the case of their ears, they had a little tube going in the ears that they just filled with string to kind of block their ears. And then I forget what it was that they used to make sure they couldn't open their mouth properly, but they were temporarily muted. They didn't kill any bats. They didn't permanently injure any bats, but they temporarily disabled each of these senses one at a time to test the bats and their ability to fly through this chamber. They figured out that the blinded bats, the ones who had their eyes closed essentially, still could easily fly about. Even if they were seeing bats, they could easily miss the obstacles and they landed perfectly normal. They had no trouble apparently navigating around the room. However, the bats that had their ears temporarily disabled and were muted, both those two groups tended to have lots of collisions. They appeared not to be flying nearly as well, and they also had trouble landing properly. They would ram into walls several times before they realized there was a spot to land at. So the The blinded bats seemed to have no difficulty, while the deaf and muted bats, they tended to have a lot of difficulty flying about all of a sudden. So that was the basics of their experiments. They had all these different little trials where they had disabled these sensory organs on the bats themselves. And now we can get into dissecting how this experiment was working. Again, we're going to first look at what's called the independent variable. What is being changed on purpose? Well, in this case, the experimenters, the two gentlemen, were changing which organs on the bats, which sensory organs on the bats were being disabled. So, were their eyes no longer working? Were their ears no longer working? Was their mouth no longer able to, be used? Which, sensory organ had they temporarily disabled? The next most obvious question, what's the dependent variable? Well, what were they measuring? In this case, it's, they had video footage as proof of this, but a very simple measurement to get out of this was simply the number of collisions that the bats had with either the walls or the obstacles / landing areas. Wherever they were trying to go, how many collisions did the bat have in a, you could say a given time frame. So now that we've identified what was the independent variable and what was the dependent variable, the next question, well, what was the experimental group? Which group were their changes made to? In this case, they actually had 3 experimental groups. You had each of the groups who had one of the sensory organs disabled, ears, eyes, or mouth. Those were all 3 different experimental groups, which means the next question is actually easy. What was the control group? Well, the control group would have been where they had just let the bats, fly around in whether it be pitch black or daytime, where none of the sensory organs had been disabled, because that's where nothing had been changed. So in this case, you have an experiment with 3 experimental groups and 1 control group for comparison. Constants. What do you think the scientists had to make sure they kept the same between all these different trials? And there should be some fairly easy things you can think of. They had to allow for the same time of flight. So you couldn't measure one bat's time at like 40 minutes and another bat's time at 5 minutes for flying about the room. You had to use the same time index for measuring flight. How long it is, I forget. I never figured that out, but I'm sure it was the same amount of time, more or less, for each trial group. You had to have the same basic obstacles in the room. So you couldn't have, increased the number of obstacles for one group or decreased it for another group. You had, you couldn't change the room as it stood. You had to have the same amount of noise volume in the room. So, it's either going to be really quiet or really loud. And that was actually something they did later play around with. Later experiments showed that when they actually turned on an ultrasonic emitter, that also screwed up the bats' ability to navigate, because they had all this excess noise that confused their navigation system. And of course, you want to make sure you're using the same types of bats. So I believe they used brown bats for these first original experiments, but they've since done tests with other bats as well to figure out how they navigate slightly differently using the echolocation. But in this first experiment, yeah, you want to make sure you're using the same types of species of bats. Same lighting conditions, whether it be dark or light, whatever you're going to do. And then the same starting location for the bat. So you don't want to start one from like the top of the roof and the other from the bottom of the floor or someplace like that. So all these things have to be held constant. That's why they're called that. And anything that changed, that they allowed to change, might make their results a bit more invalid, a bit less valid, I should say. Their results were fine. But yeah, this is some of the earliest experiments for how we knew bats used echolocation. And if you're wondering why changing the mouth organ kind of might have messed up the bat's navigation, well, not just blocking the ears, why was the mouth so important? You got to remember that bats use their mouth to emit the sound first of all, and then it bounces back off an object, like you can see here on the right-hand side, and then that response tells them whether the object is standing still, whether it's going away from them, whether it's coming towards them. But if you can't emit the sound, you're obviously not going to hear any type of echo. So yeah, by plugging up the ears and the mouth, you basically disabled their echolocation system. So that's why the muted ones also had trouble navigating about. So now we're going to get on to the hypothesis part of this. This is the hardest part. Again, make sure you've watched that video already on writing a stronger hypothesis, because again, she shows you 3 different styles. I'm going to use what's called the blended hypothesis, where I've merged all 3 of those styles that were mentioned in that video. We're going to use if, then, will result in, and compare to in our hypothesis to explain what the correct hypothesis would have been for this experiment. Now, we're doing this after the experiments. We have the hindsight, we have the ability of the benefit of hindsight. We know what's going on, prior to that. The scientists didn't at the time. They didn't really, have a correct idea per se, but this would be the hypothesis that would end up being supported by this evidence they collected in their experiments. And so you'll see it in the same basic format you see down here at the bottom. We'll say, if bats are using their mouth and ears to navigate around obstacles in flight, and you can see that's the if, what is causing what. Bats are using their mouth and ears to navigate around obstacles in flight, because they didn't have a term for echolocation at this point in time, so you can't use that. Now we're going to go with this comma, and then you're going to go with then. Then you could say by disabling, but then disabling either the mouth or ears of a bat will result in, what's going to change? The bat's navigation past those same obstacles, having more collisions. So we're going to have an increase in collisions. You could say, I said having more, you could say having an increase in number of collisions would be another way of saying this. Compared to, and we're going to say compared to what's our control? Compared to when neither of these 2 senses are disabled. So the whole hypothesis ends up reading like this. If bats, and I use my highlighter here, if bats are using their mouth and ears to navigate around obstacles in flight, then, let's say, by disabling either the mouth or ears of a bat, you could say this will result in the bat's navigation past those same obstacles having more collisions. So we expect there to be more collisions when the mouth or the ears are disabled compared to when neither of these 2 senses are disabled. And that would have been the correct hypothesis. Because again, you didn't see more collisions when the eyes were disabled. They were perfectly fine there. But again, this is just me showing you a demonstration of using this blended hypothesis format. And you can see why it works so well. It is wordy. Yes, I know, it is long. But it comes out and it tells you exactly what you expect to happen, what it's going to result in, what you're changing, what's going to result in, and who you're comparing it to by the end. This is why I call it a blended hypothesis, compared to that video with the girl talking about the 3 different styles. So that's the experiment. And make sure this makes sense. Again, if you need to go back and watch the hypothesis video to understand where this blended hypothesis is coming from, or watch the video on the bat experiment itself to understand what they were doing, then fine, do that. But understand, we are just dissecting the different parts of this experiment and how it played out. Again, there will be 2 more in this series of videos, so make sure you watch both until you are comfortable understanding how we get each of these different aspects of the experiment, okay? If you have any questions, though, let me know in class. I'll see you guys